Hi student, in this video I am going to explain you about the pollen grain. He has given pollen grain heading only in that NCERT book. But under that book, under that heading, he has given the structure and formation both together. He has given formation, he has not shown, he has not written, but he is shown in the diagram. That also I will explain. And it is very important for the NEET because many bits can be created from this, that is the pollen grain formation and structure. So it is most important for the NEET. Now you see, first the pollen grain or pollen grains are represent male gametophyte. Why it is called as male gametophyte? The haploid cells which are producing the gametes, if they are producing the gametes, then it is called a gametophyte. Because how it is that you will come to the in the discussion I will tell you. So gametes are not directly produced. Some haploid cells used to produce gametes, and these haploid cells which are producing the gametes, that is called as gametophyte. Now, whenever we are seeing the pollen grain, you collect different pollen grains and observe it under microscope, then you will can see amazing, so many types of the designs, means that is they will be the having the smooth wall and rough wall and up and down, so spiny like structures, different colors, many architecture can be seen on the pollen grain. Why? How it is? It is formed by the Exine. This Exine is the outer layer of pollen grain. And next, here, whenever we are seeing pollen grains, pollen grains has two layered wall. The hard outer wall is the pollen grain has two layered wall. Around it, it is two layers. It is to present. It will be protected by two layers of the wall, cell wall also we can call. That also I will explain you. And the hard outer layer, the out of the two layers, outer layer is very hard and is exine. It is called as a exine and is made up of the sporopollenin. This is most important. Sporopollenin he asked once in the last 5-6 years. And why it is that much important? Because the sporopollenin is the organic material and it used to have, it can be withstand, tolerate that material which is present in the outer layer of the pollen grain. Outer layer of the pollen grain is sporopollenin, and sporopollenin is having the resistance to the temperature, acid, alkali, and any enzyme cannot digest this. Such type of the material, it is made up of lipids and phenyl compounds, that is out of not necessary. Sporopollen is organic material and it can withstand, means tolerate high temperature, strong acids, alkali, or any enzyme cannot digest it. So that much hard material it is. And why it is that much hard? Because the pollen grain is very small particle. It will be whenever the shed, it is to come out. And then what happens? It will be exposed to the environment suddenly. From the anther, it is coming out. So it must tolerate the temperature and all the things around it. And it used to fall on some other places also, and it should be withstand, means it can be stayed for many days also. This so such type of the sudden exposing to the environment, it must be the very hard. That hardness is what it is. The sporopollen is very hard material which cannot be destroyed by the temperature, acid, or the enzyme. And here, that what happens because of that one, So pollen grains are preserved as fossils. So it cannot, temperature cannot do anything, acid cannot do anything, alkali cannot do anything, and enzyme cannot digest this thing. Because of all this nature, what happens? So many thousands of years back, repairings also used to present that, we used to call it fossil. So the fossils, pollen grain fossils are available because of, we will ask, outer layer of the pollen grain. Pollen grain fossils are available because of outer layer of the pollen grain, that is the made up of the sporopollenin, which can be withstand all the things. So for that is the reason. This is also very, very important need bit, because you cannot expect how he will give, in which direction he will give, but we have to understand the concepts. Then any angle he has, he is to give the question, he can give the answer. Now Exine is very hard material. Okay, so it is covering the entire pollen grain. So if the hard material, if it is like that, the pollen, from the 8th class, 9th class also you are studying, that pollen has to produce pollen tube. The pollen tube has to come out from the pollen grain. For that one only, this Exine is absent here and some places, and that their sporopollen won't be there. So it is leaving the apertures, means small holes here and there, means that it is not continued. Exine is not continuously occupied the outer, as a outer layer. It is to leave here and there some apertures. These apertures are called as germ pore. The use of the germ pore is, the pollen tube has to come out from the pollen grain at the time before the fertilization, means it has to come out and it has to travel through the style and it has to reach the embryo sac. Those things will come later. First it has to germinate and pollen tube has to come out. If it is continuously present, it cannot come out. For that only apertures are left in the Exine. And Exine also is important for the formation of horticulture things on the pollen, that also were given in the starting one I told. And next, whenever we are seeing the inner wall of the pollen grain is called as intine. Means outer wall, two wall layers are present around the pollen grain. Outer layer is the Exine. Intine is just behind the Exine. Outer wall, one more wall is to present, that is called as intine. And intine, how it is? It is the thin wall and made up of outer wall, sporopollen, it is that Exine. Inner wall also he has given the material. All these things are need bit, remember it. And this intine is made up of cellulose and pectin. Cellulose and pectin are present in the intine. Sporopollen is present in the Exine of the wall. That's a pollen wall. That we can say cell wall also. That is different. The discussion is different. And thin continuous layer made up of cellulose and pectin. Here. Now we'll see. Now inside the two walls, what is present? That present it is cytoplasm. So cytoplasm, anywhere cytoplasm is there, cytoplasm is surrounded by the plasma membrane. That only is telling the cytoplasm of the pollen grain is surrounded by a plasma membrane. So plasma membrane, upon the plasma membrane intine, on the intine Exine is present. Exine is made up of what? An intine is made, and plasma membrane. These are the things you have to remember it. And next, this is the external structure of the pollen grain. Pollen grain is to form in the maturation only. For that only, formation is important. In the formation also, now we will see formation of pollen grain. how it is formed. He has shown very clearly in the diagram series. And why the diagrams he has given, how do you have to remember it, because the concept is there, some concept is there in that. That we will see now. Pollen grains formed by pollen grains forms by the further changes of microspore. Microspores are formed in the microsporangia. In the microsporangia, centrally it is have the, that is microspore sporogenous tissue. The each cell of the sporogenous tissue will be, act as the spore mother cell, and it will take the reduction division, meiosis division, and forms 4 haploid spores. But these haploid spores, they are here. This is the haploid spores which are formed from the mitotic division of the spore mother cells inside the sporangium in the sporogenous tissue. Here the microspores, 4 are haploid spores are produced from the spore mother cell by the reduction division. They are with the, after reduction division, 4 are formed. Immediately it cannot be separated. They used to form the cluster that we used to call a microspore tetrad. This is the microspore tetrad. But this textbook wrongly has given, but he has written in the sporogenesis correctly. See, enlarged view of pollen grain tetrad. This is the wrong one he has given. And next what he has given, stages of microspore maturing into a pollen grain. From here, whatever the stages he has given, then it is microspore, that is the B, he has given stages of microspore maturing into pollen grain. He is telling very clearly. Then how it will be the spore tetrad, pollen tetrad? Wrong or not? It is wrong. But he has given one place correctly, that is the microspore tetrad. He didn't mention there as the pollen tetrad. Remember the point. Such type of things only he will catch in the NEET. Remember it. Now what happens? This pollen microspore tetrad will be dehydrated. Because of dehydration, it will be separated. Spores will be come out. The spore used to have one single cell. Microspore is a single cell, single nucleus. This spore, microspore, used to take the mitotic cell division. Remember it. He has shown this. This is the whatever the, it has shown the division also. Spindle fibers and all the things. That is the mitotic cell division. It taken. It is a haploid spore. It taken the mitotic division and forms the 2 cells. When the mitotic division has taken, the 2 cells generally equal size, but here the main thing is they are dissimilar. This is most important for NEET. One cell will be very big and the other cell will be the very small. The big cell is called as vegetative cell and small cell is called as a generative cell. How much small it is? It used to float in the cytoplasm of vegetative cell. That he has written in that book also. So it means it is on surface, it is stretching, means it is floating on that. So it used to float on the cytoplasm of the vegetative cell. And both the cell, the vegetative cell, inside the vegetative cell, generative cell is there. After mitotic division, the daughter cell used to present inside the other cell. What is that? If you ask what you will give on, this is the thing. After mitotic division, 2 cells are formed, but the one of the cell will be very small and it is to present inside of the other cell, that is the pollen grain. This is also NEET bit. Remember it. So like that you have to prepare. And next, these are some variations are present between the vegetative cell and the generative cell also. Vegetative cell is very big and it used to have the vacuoles. He didn't mention in the text, but he mentioned here. He can ask. It is having the vacuoles and cytoplasm rich with nutrients. Means whatever the required for the growth of the pollen tube further, it will store in the vegetative cell. Food material will be stored in which cell of the pollen grain, if you ask, that is the vegetative cell. Remember it. And it is having one more peculiar character that is given in the book also. That is, it is having large nucleus. Nucleus size at the mitotic division has taken. Vegetative cell and generative cell both must have same size nucleus, but it is not. It is vegetative cell nucleus is big size and irregular shape. Nucleus means circular, but it is irregular shape. Large nucleus used to present inside the vegetative cell. This is most important for the neck. And next, this is irregular. But generative cell nucleus and vegetative cell nucleus both are generated by the mitotic division of the spore only. Remember this point. How many differences are there? And vegetative cell used to have the rich of food material, nutrients, and those nutrients are useful for producing of the pollen tube. At that time, it required a lot of energy. For that one, it is having the lot of food material in that. And generative cell is spindle shape. This also need bit. It is in the spindle shape, and nucleus used to present inside, and thick cytoplasm used to present, dense cytoplasm used to present in the generative cell. All these things he mentioned. Now is it necessary to separate the formation of the pollen grain or not? Yes, it is. Pollen, this is that microspore cell, microspore, and has taken the one mitotic division and formed two cells. Two cells are having the some dissimilarities. Now you tell this is the whatever the outer layer and inner layer of the whatever we read in the pollen grain. They also can be called as cell walls because they are the covering the entire pollen grain. Entire pollen grain is nothing vegetative cell because generative cell is present inside. Remember this, all these things, remember it, because the questions how it is created, that we cannot say, but the concept, if you know how he created, you can understand. So these are the things. Now we'll see the same things in the formation of pollen grain. Pollen grain forms by the further changes of microspore that we shown. Each microspore take mitotic cell division and forms two unequal cells. One is the big cell and one is the small cell I discussed. And the big cell called vegetative cell and has abundant food reserve vacuole with irregular shaped nucleus. Why these all the sentence I only created, I only write on this board. One facility is there. Whenever I am writing with my handwriting, it will be converted into the printing letters. These all the sentences are my own. So here, why I am telling, you can write if you want to write notes like thing, you write it. Because the persons who are going to give examination, CBSE examination in the 2027, they are having different types of the questions. They are equal to the questions are equal to the NEET only. So descriptive questions, for that these notes will be useful. Prepare the notes from this. So here the big cell called vegetative cell and has abundant food reserve vacuoles with irregularly shaped nucleus. I discussed already, I shown also. The small cell called generative cell and generative cell and floats in the vegetative cell because it is a small cell inside the other cell. That also I told the thing it is. Next, whenever we are seeing the generative cell is spindle shaped and has dense cytoplasm and nucleus. The shape of the cell also has given very clearly spindle shape, irregular nucleus and vegetative cell. All these are neat bits. Please remember it. And now the matured microspore called as pollen grain. Now the thing is here very clearly told in the textbook also by the microspore tetrad separated, spore came and spore and microspore as developed as the pollen grain by taking the mitotic cell division, and in that division also it is formed the two unequal size cells. One is the big size cell that is called a vegetative cell, and small cell generative cell, and generative cell used to float in the cytoplasm of vegetative cell. All the things you have to remember it properly, because you see how many types of the questions you can create. For that one I am telling this much detailing. And here he has given very clearly enlarged view of pollen grain tetrad. Completely wrong it is. It is a microspore tetrad. He has written in the microsporogenesis, but he has mentioned here very wrong. And stages of microspore maturation into pollen grain. It is correct. For that only I separated the heading. Is it correct or not? Correct it is. Now we see here. So in this way the pollen grain is formed. Now the matured microspore called pollen grain. Is it correct? Yes, it is. You can ask in the matching anywhere statement. You can give anywhere in the NEET also. Now you see. So when the pollen grain is formed, 60% of angiosperms pollen grain are shed at 2-cell stage. This is the 2-cell stage. So more than 50% means 60% of the means when the pollen grain is coming out from the anther, at what cell stage it is to have? Generally it is to have the 2-cell stage. That is the vegetative cell and the generative cell. 60%. Remaining 40%, what it will do? What they are doing? The generative cell used to take one more mitotic cell division and forms two male gametes. That whatever the generative cell is there, generative cell means which it is generating? Male gametes. So male gametes are formed by the mitotic division of generative cell. Now you tell me, if it is shedding at 3-cell stage from the pollen mother cell to formation of 3-cell stage, how many meiotic divisions has taken and how mitotic division are taken? First meiotic division in the microspore mother cell will be taken one, and microspore here whenever we are seeing, it has taken mitotic cell division and forms 2 cells. So one mitotic, and 3-cell stage one more mitotic cell division is taken. So 2 mitotic divisions, 1 meiosis division. You will ask any questions in this. So here 60% of the angiosperms pollen grains are shed at a 2-cell stage. Remaining 40%, whatever are there, they will shed the remaining shed in the 3-cell stage because of the generative cell divided mitotically and forms 2 male gametes. Understand? Now the pollen grains are very small size. It is generally 2 cells or 3-cell stage only it is 2, and it will be spreaded into the air also because of the where it will be anemophily. Means the agent, whenever the air is there, anemophily we used to call, that will come later on. At that time it used to release in the air. And whenever we are breathing and going that side, the pollen grain will enter out inside and the other respiratory tract, and it will give some allergy. That only the pollen grains of many species cause severe allergies and bronchial afflictions in some people. Some people used to get the irritation of that pollen grain. Why? The pollen grain is to have some peculiarities on its exine. Because of that one, or it may be germinate because of the wetness in our body. Pollen tube used to come, it used to irritate something, it used to happen like that. So causes severe allergies and bronchial affinities in some people, not in all. Example, Parthenium. Parthenium is also called as Congress grass. It came to India. It is not Indian. India, it is evolved in not India. Some other countries, whenever we are importing the wheat, along with wheat it came. So we eaten the wheat and thrown the seeds of the Parthenium, Congress grass. It become a popular problem to entire India because it is occupying and it is giving the irritation, small allergies, all the things it is giving. That is the thing. Now, one more thing he has given. Now, already I told, vegetative cell is the big cell, rich of nutrients. So, this is why I am telling pollen grain rich of nutrients. So, they are used as food supplements. We will eat whatever the food material is there. That food material is meant for the pollen germination and pollen tube formation, but we are eating those pollen grains also nowadays in the form of tablets and syrups because it is having a lot rich of food material, sugars, and all the things are present in that. Mainly athletics, they used to do race horses and as per other whatever the purpose we are using this. Now the next thing is viability. Pollen viability. Viability means why it is viability, why it is required. The pollen is shedded from the anther and it came out. It may reach on the stigma of the carpel, but in that time that agent may come or may not come. It may come very late. For example, honeybee, it came very lately, but already the pollen anther is shed, means it is broken and the pollen grains is ready. So at that time, the delay time is there. Because of this delay time, it will lose its viability, means its survival, means it cannot produce pollen tube if it is dies. If it is viability means how much time it is having the life to produce the pollen tube. And the pollen tube, when it will produce it, that it is to go and it will fertilize the egg and all the things. That is a different one. So the viability of the pollen grain used to vary. It is to vary. Different species in different timings it used to have. For example, very less time means shedded pollen grains viability is different in different species. In rice and wheat, it is 30 minutes only. This is very important for need. Very less time. And next, in some members of Rosalis, legumes, Solanaceae, it is few months it can. The pollen grain comes also, it will be survive for few months. That much it is. It is having that facility. This years twice. Rosalis, legumes, and next is the Solanaceae. Twice he has 21, 23 years, and 18 also he has. But in different ways he has. And such type of the viability, it may be the 30 minutes, it may be the few months, some of the species. So variation. Now the pollen banks, they started like the sperm banks like that in the animals. Here also pollen will be stored. Why? Because of this, whatever the plant breeding purpose. They want to do something. Scientists at that time, the plants are not they may be not in the flowering season. They want that, they want to do some research on that one. So they will get the pollen grain from the pollen bank. That must be in the living condition, means viability must be there in that one. So for that purpose, plant breeders, they started pollen banks. So they, in that one, the pollen grains can be stored years together without death. So here the pollen banks, in the pollen banks, pollen grains stored for years in the liquid nitrogen with which is at minus 190 degrees centigrade. Liquid nitrogen means all the sperms, whatever the sperm bank is there, that also one liquid nitrogen only. It is to have the minus 196. He asked this one once in the 18 or something. So questions they asked in this way, but in the formation of the pollen grain, how many questions can be created? And the Exine and Intine used to cover which cell. If you ask, what you will say? Pollen grain. Pollen grain, if is not given, vegetative cell it is to cover. Concept you should understand first. So this will be stored. This is about the formation and structure of pollen grain. In next video, I will explain you about the megasporogenesis. So I think if you feel this video is good, please immediately give the like mark. And all the videos will be explained like this. From now onwards, every day or alternate day, I will give such type of information to you through the videos. So please subscribe the channel. Thank you.